Thank you. Thanks for the invitation. Yeah. This is a project that I've been involved in, or really a series of projects I've been involved with Matt Bennett's lab at Rice University for a while, and for maybe 15 years now, continually. And the question I'm going to let me just kind of tell you what the who the people are. Oh, let's see. Does this work? Okay. Yes. All right. So just because I usually forget, so I'm always usually put I mentioned the people who actually did the work first. So again, as I said, this is work with Matt Bennett's lab. Matt is actually a physicist by training, but he went off into experimental synthetic biology, and but it makes it really easy to communicate with him. And so my part of my group typically does the modeling, or it's really joined, and Matt's group does the experiments that you'll see come from Matt's group. And here are all the people who've come through our groups over time, including some people who are still at the University of Houston who work with us. So what I'll tell you about is really kind of a couple different aspects of the problems that we've been looking at. And one of them is the oscillations and how they're affected by orientations in microfluidic traps. And I will give you a little bit more detail if you haven't seen microfluidic traps. I will tell you a little bit more about what they look like and why we're using them. And next I'll talk about pattern formation and bistable populations. That's going to be maybe a shorter part. All right, and so the motivating the motivating question really that's driving our research for the last about 5-6 years or so is the following. So how do you get different strains of bacteria to communicate and exhibit complex emergent behaviors that really you cannot understand if you look at the individual strains on their own? Okay, and so by strains here, I'm going to mean they're actually going to be E. coli. They're all going to be E. coli. They're going to differ. The strains are going to differ in a plasmid, or they usually contain about several copies, about 20 to 30 copies of a plasmid, which carries a particular genetic circuit. I'm not going to go very much into the detail of those genetic circuits, but that is really going to be the main difference between the bacteria, right? They're going to have a really small genetic circuit that is different between the different strains in the bacteria. And that circuit also typically carries, or the different strains of bacteria also express a fluorescent protein that's different between the strains. So when you see the different colors in the movies I'm going to show, typically cyan and yellow, that means that you're looking at the genetically different strains. Again, genetically means that they're the main part, these little loops of DNA are just different between the two different strains. So this little circuit that's been inserted by the synthetic biologists, by our collaborators, is different between the different strains. Okay, so how do the question that we started with really down this road was this 2-gene dual feedback oscillator. This is something that Matt's worked on as a postdoc, and it is a robust oscillating circuit. And so what you see here really is a circuit that is typically inside that was originally inside a single cell, right? So this was essentially 2-gene, one which activates the other, the one that also is, when it's on, it also it's co-expressed with a cyan fluorescent protein, and the yellow one is co-expressed with a yellow fluorescent protein. And you can show that this genetic circuit, again, activation and repression, self-repression, and mutual repression and self-activation, and activation of the partner gene, it oscillates robustly. So that was shown, right? And so the first thing we did was, suppose you take this gene, right, and you put it in one population of bacteria, and take the second gene and take it in the second population of bacteria, and then couple them via extracellular signaling, right? And so the cyan molecules, the cyan of bacteria, right? So this particular strain, when active, expresses a signaling molecule that activates all the other bacteria in the population, right? And both the yellow and the blue, and it's similar for the yellow, but it's repression, right? And so that's kind of the idea of this particular of this construct. Again, take this from inside a single cell, and create this at the level of a population, right? Via signaling, via extracellular signals, rather than via intracellular signals, that was originally done in the original papers, that are about 15, 16 years old now. All right, what you see that this, what we were able to show that this actually robustly oscillates as well, right? And so you get a robust oscillation just at a population level, just as you do in the level of a single cell, and I'll show you this in a moment. But we'll show what we'll I'll skip right into kind of the more interesting part, where there's actually looked at also what happens in spatially extended consortia. And what you'll look at, just to briefly describe, is an extended consortium that's grown in a microfluidic trap. So this is the trap, and it's about a single layer. So it's constructed in such a way that what you're looking at here in this trap is a single layer of cells, right? And so what there's actually no multiple, I mean, there's sometimes overlap. There's about 1 micron thick, this trap inside the trap, so that the cells are trapped there, and you're looking at a single layer of cell, a monolayer of cell, and there's media flowing to the sides which provide nutrients to the cell and take away waste, and as the cells grow, they're growing into these media. So this is looking at, so if you take a cross section, oops, let me go back. If you take a cross section right here and you look at it from the side, you see the flow is going into, carrying into the screen here, and carrying away the bacteria that grow outside of the trap, and then removed and flushed out, right? And so the population inside the trap is roughly constant over time, because once the trap is filled up, but the bacteria keep on growing and displacing each other and moving into the side channels and being carried away, right? And so inside the trap, the bacteria grow, and they're exactly consist of the 2 strains that I've just described. They signal to each other, and what you see in those cases is these temporal oscillations, right? And so what you see here is about 1 million bacteria growing in this microfluidic trap, signaling to each other and oscillating because of this genetic circuit that I've just described, and via also this extracellular signaling. Right. And so what's been really important in this, and so what we've done in this paper, I'm not going to mention it, is essentially what you see is that the signaling in these traps, the signals propagate about 100 microns. The entire trap is about 1 millimeter. So the signal propagates from let's say, from a column of bacteria, the single column of bacteria over here will propagate about 100 microns to the sides, but the entire trap is, if you look at the oscillations in the entire trap, is synchronized. So what we did with Jae Kyung about 5 years ago was to then try to understand how it's possible that you get essentially synchronous oscillation in the entire trap, while when signaling is essentially just local. So how is this coordinated? And so this kind of a longer story, I'm not going to go into it. The other thing that we looked at here is, why do these bacteria organize? If you look at them, right, they organize roughly in this columnar structure. They're not, it's not exactly right, but it's roughly, they're pretty organized in a way that's kind of perpendicular to the channels that are set. Remember, on the top and the bottom, there's a channel where the bacteria go out and are being then carried away by the flow, into the media. All right, and so this is the question that we asked. So there's a couple of questions that we asked, and that was actually pretty important for the synchronized activities, the spatial organization, because if you change the trap and there's a lot of tumult and the bacteria kind of grow in all possible directions, the synchronization is much weaker. So this is actually also important for the spatial organization of bacteria, is also important for the oscillations to be maintained. Okay, so this is again just amplifying a little piece over here. Again, the structure is not, the organization is not perfect, but you do get roughly this columnar organization, these columns of bacteria inside these trap, and they're perpendicular to the long side of the trap, right? And so the question was, why this happened? So Bhargav Karamched, who's at FSU right now, created this spatial Moran model to kind of try to capture the main potential mechanism behind this. And the idea here is that you have essentially, like in a Moran model, you have bacteria. Now they have a spatial orientation which is either horizontal or vertical. If a horizontal cell divides, it will displace cells to the left and to the right. And if a vertically oriented cell divides, it will displace the cell to the top and to the bottom. Cells at the boundary are going to go into the channel and be carried away and disappear for the population. This is how you guarantee, this is how you keep a constant population. And this is essentially a simple lattice model. There's a lot of people who worked on this, mostly in terms of trying to understand cancer modeling cancer or different tumors, spatially organized tumors, but also in bacteria, in the microfluidic traps, bacteria microfluidic traps. So if you do this, you have to add one important thing, and that is that displacing cells in the direction where there's more cells between, let's take this red cell over here. We're going to have, if you displace a cell in a direction where there's more cells between the cells and a trap, that is less likely than displacing a cell to the bottom here, where there's fewer cells between the dividing cell and the channel, right? And so it's less likely for this cell, it's less likely to have a descendant to the top than to the bottom, right? Because it's easier if you think about it just physically, it's easier to push out the cell than to push out the cell at the top, simply because there's more cells to move out of the way, if you wish. And this is not arbitrary. In fact, there is experimental evidence that has come out since from Simona Pigolotti's lab that shows exactly how this probability changes as a function of location inside a trap, and it's a roughly linear change in the probability of displacement to the left and to the right. And these lattice models have been verified as well to be relatively accurate descriptions of what happens in these microfluidic traps. All right, and what you find is that in this case, you get that particular point, this fact that you have this difference in displacement really gives you this nice spatial arrangement that we talked about before. But if you do not take this difference in probability of displacing cells to the top and to the bottom, displacing the cells in the direction closer to the boundary, you do not get this nice arrangement. So this was one of the things that's kind of necessary in these lattice models. Okay, what's also nice about this lattice model, again, I'm showing kind of a minimal of math here, is that you can write down very nice mean field equations for it, essentially a population model mean field equation. And surprisingly, even though there's correlations between neighbors, the mean field models actually work relatively well, right? And so if you look at there's a single realization of the fraction of vertical cells. This is the mean field model for the fraction of vertical cells. This is the blue line is averaged over multiple realization, and the black line is this mean field model. So, even though you have, relatively very noisy behavior in single simulations, on average, the mean and correlations as well, actually really quite strong correlations at the boundary. The mean field model surprisingly works really well in this particular case. All right, and so but you do lose a bit, right? You lose quite a bit when you look at these lattice models that tell you something, but they don't tell you enough detail. So what we've did also is to look at agent-based models that capture some of the mechanical interactions that I've just described. And you can think of the agent-based model as essentially a drawer. You have essentially two halves of the cell, let's say green and a red part of the cell. So there's two different halves of the cell that are being pushed apart via a spring whose length increases, whose relaxation length increases, right, as the cell grows. And so when the string is relaxed, the cell would relax to the cell length will be determined by the length of the string. But if there's other cells in its neighborhood, the spring is being compressed, and the cell is essentially it does not achieve its kind of the relaxation length. But then once it goes into a less crowded environment, it can extend again, right? And so you can make this a little bit more realistic and also provide mechanical feedback to decrease the length, to decrease the rate at which the spring grows if the cell is in a constrained environment, which is a little more realistic, but you get similar results. Okay, and then you put these into a agent-based simulation, you get very similar arrangements to the ones that you see experimentally. It's a little bit difficult to validate these, but we tried in different we tried to simulate these in different trap geometries, and you see very similar arrangements to the ones that you see in traps as well. So this is growing a colony in a trap takes about a few hours, no, not a few hours, a few days, right? And so this is a lot simpler to experiments with these than with cells in traps. And so what you find here is that if you have multiple strains growing in the same trap, you get two types of displacement, two types of ways in which one strain can displace the other one. This is important if you want to have a consortium. You don't want to displace the other, the other strain or one of the strains, and will be left with only one. So it's important to kind of look at the ways that that strains can displace each other. One way is via these invasions. And if you see here, you can see the start of this invasion where these columns of one strain are being invaded by the cells of the other strain. And the other type of displacement you see is what we call bulk displacements. And this is when you have one strain that has slightly smaller cells that are a little bit more round than the other strain. What happens is that these cells, again, you can quantify this, I'm just kind of giving intuition here, that these cells are more likely to grow in have a horizontal component in their growth and thereby displace the other strain, right? So these cells, the orange strain in this case, has a smaller aspect ratio, is thereby also more likely to go in the horizontal direction. And this horizontal growth is displacing the blue strain. The blue strain mainly grows in the up and down direction and therefore has no horizontal component in displacing it. All right. And so again, you can try now play games with this. For instance, in this case, we have a oscillator strain. The one of the strains is smaller than the other one, but it's only small, has a smaller aspect in as the other one, but it has a smaller aspect ratio while it receives a signal from the other strain. In other words, if the strain 2, let's say, stops receiving a signal from strain 1, then it will go back to a longer aspect ratio. So you get essentially a negative feedback mechanism here, and you get a nice oscillation between the two strains. The smaller strain, right, this is the smaller strain, and then now it turns off, it becomes longer. The purple strain is now the shorter one because it receives signals from the blue one, and the two just keep on oscillating. And you get essentially a relaxation oscillator between the two. Okay, so we're still trying to build this one in a microfluidic trap. We kind of think that there is genetic machinery that will allow the cell length to change, but we haven't gotten to that point. We haven't gotten it working yet very well, especially with two strains in this particular way. So this is more at the theoretical level right now. All right. Okay. So the last thing I'm going to talk about is a synthetic toggle switch. This goes back to essentially the dawn of this type of synthetic biology. This paper by from Gardner, Tim Gardner and Jim Collins is from Jim Collins's group in Nature, would have constructed a genetic switch, right? So essentially, these are two genes that inhibit each other, and essentially you get a bistable switch where either one of them is one is being expressed or the other one is being expressed, right? This is an old paper by now, and it's been repeated in many different situations, right? And so you can change, then you can either activate one or repress the other by introducing an inducer, IPTG, or via heat. In their original model. In our case, we only use an inducer. So this was the original mathematical model that they put in their paper. It's very simple, right? So you can see that the two repress each other. The larger the more one is expressed, the smaller the rate of expression of the other gene. Essentially, this is kind of sophomore level model but it was really the motivation for this particular circuit, right? And so what we did, again, the same question as before, take that particular circuit, right? And so this is the implementation of that circuit. Again, this is the yellow fluorescent protein and the cyan fluorescent protein that are being co-expressed with these two genes that are repressing each other in a single cell, and take that to a population level, right? And so these molecules that repress one another are now the proteins that are now replaced by signaling molecules that propagate extracellularly, enter the other cell, so that when the cyan part of the circuit is being expressed. So essentially it's a little bit more complicated here because we also have to express this signaling molecule. Then it will repress all essentially the yellow part of the circuit in all the neighboring cells, right? So this extracellular signal will propagate and it will repress the yellow part of the circuit in all the cells, and all the neighboring cells, right, as far as the signal propagates. And the same thing for the yellow, right? The yellow part of the circuit, when on, is going to repress all the blue part of the circuit right here, right? And so essentially it's taking this to the population level, right? Exactly as we did with the oscillator before, but this is it's just a different geometry, or say different architecture. Right? The difference here is these are not going to be grown in microfluidic traps. And I have 2 minutes or 3 minutes. Yep. Okay, I'll wrap up soon. So these are not grown in microfluidic traps. They are grown in colonies, right? And the colonies are a little bit different than traps. So, let me just show you first. I thought they were going to be. So these are little colonies that are grown in agar, and I'll show you the geometry of them. They're not monolayers, and that's going to be important for the model. Okay, and so what you find is very different behavior depending if you grow a conventional toggle, right? There's no extracellular signaling, and a quorum sensing toggle, one with extracellular signaling, where neighboring cells are able to communicate with one another. And so what we have, we built a small PDE model, right? This is, again, the model of the circuit with signaling diffusion in the case there's a quorum sensing. In the case there's no quorum sensing, there's only essentially expression within the cells. This is a very, very simple model, the one, pretty much, again, not much more difficult than the model that was proposed by Jim Collins's group, except for the presence of the signaling molecules over here. Again, what you have to take into account is this growth, is this spatial geometry of a colony. And you find that the colony essentially, if you look at it as a cross section of the colony, kind of looks like a pyramid, right? And so this is like half a cross section of a colony. And the importance, so this is again the spot of bacteria growing in agar. Agar is at the bottom. Most of the growth is here towards the bottom. This is kind of the interesting thing. These cells at the top of this pyramid do not grow very quickly. The cells at the bottom are the ones that are in contact with the agar that get the most food, and they're the ones that actually grow. If you do this and you simulate the PDE on this particular growing domain that's important that it's growing, you find that you get exactly the types of, if you match the parameters, you get exactly the types of patterns that you see experimentally, right? In a non-quorum sensing toggle, so not quorum sensing, so the original toggle, you see these blue cells, right? So again, you start in the yellow with mostly yellow cells. These are the ones. The toggle is a little bit asymmetric. If you start, the yellow cells kind of tend to be more dominant, tend to be on. But as you grow, the blue cells appear at the top, and you get a ring pattern, essentially, because if you image the colony from the top, you see the blue cells, they're on the top, and the yellow cells are at the boundary because there is no blue cells. So essentially take this and spin it around, right? This is half the cross section of the colony, right? And so if you merge the yellow fluorescent and the cyan fluorescing cells, you get exactly the pattern you see in the experiment. If you do the quorum sensing cells, right, a quorum sensing cell colony, you see something different, or you see that if you fast forward, you see that the yellow cells, the yellow expressing cells are in the middle, and the blue expressing cells are at the boundary, right? And that's again something that agrees with the experiments. The one thing that you see that's very different is that in the non-quorum sensing circuit, the blue cells, the cyan cells are at the top and the yellow cells at the bottom, while at the quorum sensing circuit, the model at least predicted that you have yellow cells towards the center and blue cyan cells towards the boundary. And so that was a prediction, and it took us quite a while, but Barbara, who was the experimental collaborator on this, was actually able to cut these colonies and fix them and cut them and essentially test this hypothesis. And you see something very similar, right? So you see it's not as strong here, but you see that if you cut the colonies across, that in a conventional toggle, you see predominantly blue cells at the top and yellow cells kind of towards the bottom. And in the quorum sensing toggle, you do see the blue cells towards the sides and the yellow cells towards the middle, right? And as the model predicted. All right.
So, Crystal?
Yes. Yeah.
Oh, you're wrapping up. That's great.
Yep, I'm wrapping up. Yes. All right. So so what I showed you is kind of a group of different types of models that we used to approach this problem, understanding spatio-temporal dynamics, agent-based models, lattice models, ODEs, and so on. And I view these kind of as different pictures, kind of the same object, right? Of the same problem, right? And I think that you really, if you want to understand these, if you want to get at the mechanisms, I think all of them have their values. And I think we really need to take an approach where we combine these different approaches rather than using a single approach to trying to understand these complex biological systems. And these are actually relatively simple compared to the ones that I think most of you are looking at. All right, and I'll wrap up, and I think that's my last slide. All right, thank you.
All right, let's thank Krzysztof for the very, very interesting talk. So if you have any questions, you can mute yourself or put your question in the chat, whichever way you like to work on. So right now I don't see any questions in the chat. So Krzysztof, would you mind bring your slides back up a little bit?
Yes.
So I think it's very interesting that last, so this result. So could you provide some insights on why the two cases generate these two different patterns?
Right. Yeah, I probably went through this a little bit too fast, right? And so effectively what happens is that this is a colony, right, growing on top of agar, which I haven't shown here. That's the bottom. Below is the agar. And the agar actually has, in the agar, there's an inducer that preferentially switches the cells into the yellow state. All right. Okay. And this is what you see effectively is that but it's not, and the inducer actually degrades over time. Right. And so there is a degradation over time. So you start with mostly a yellow colony, right? Because effectively, but the concentration of the inducer essentially slowly degrades. Oh, this is the previous one, right? And you get these, cells, these blue cells. Effectively, then these blue cells are essentially because of the inducers there. You originally preferred a yellow state. As time progresses, the blue state, the inducer degrades, and you effectively switch to the blue state, right? So yellow state first, inducer degrades, you switch to the blue state, right? And then it goes from top to bottom because the inducer diffuses from the bottom to the top, right? And so this is where the concentration on the top is where the concentration of the inducer is the smallest. If you look at in this case, now you have coupling, right? And so there is a positive feedback between cells that are in the yellow state and cells that are in blue state, right? And that's actually better say is a negative there's a repression. Cells that are in a yellow state will repress cells that are in a blue state, right? So you get these large regions of yellow cell states, which essentially repress. They don't allow cells to switch into the blue state. Now, as the colony grows, again, the inducer decays, right? And at the very tip of the colony, where the repression is the smallest, at some point the repression becomes small enough to where the cells can flip from the yellow to the blue state. And once they're in the blue state, they repress the yellow state, right? And so you get this pretty sharp separation. In fact, this boundary moves back a little bit very slowly. And the prediction would be that if you grow this colony over time, they would all eventually flip to the blue state. But it takes a long time, probably longer than what we can do with an experimental setting. Does that explain it?
Yeah, thanks. So in the first scenario, so you have the yellow all the way to the edge.
Right.
So is it because the degradation, the decay of the inducer is rather slow, so you still have sufficient
Yeah,
amount
yeah.
of inducer
Yes.
in the
Right. Yeah,
okay.
yeah,
So
yeah, yeah.
like in order to get blue actually on the edge, you need that sort of quorum sensing effect because the inducer decay itself is not enough
Yeah, no.
to make that happen.
You definitely need a quorum sensing effect. And there is a slight difference, what actually is a little bit hidden. There's actually a little bit of a difference. These are not quite the, non-quorum sensing and the quorum sensing architect. The cells are not quite the same, right? Because they're the genetic circuits are a little bit different. So you have to change the parameters. It's not exactly you just add the you just add the signaling molecules and that's it, right? So you have to change things a little, the parameters a little, but not too much, right? I mean, they really are quite close in many other respects.
Okay, sounds cool.